Okay, so this is going to wrap up the information that we have as it relates to chapter 1, and we're going to be talking about biology as a discipline. So how can we use the scientific process to test and answer biological questions? So, first question, sounds simple, right? What is science? This is where we have observation. So we're going to be talking about things that we see. We're going to be identifying those items and then performing different types of experimental investigations and using, our theory of things that we know to explain what's happening in the world around us. Science is going to be an objective way of collecting knowledge. This is really important, and we'll talk about what it means to truly be objective, because that is the brilliance of science, right? We don't want opinions, we want the facts. That's how science is able to be run. So with our method, we're going to kind of take it back to something you may have seen before, but we're going to make sure that everyone's on board and understands how these components work. So we'll be discussing observation and how observations can lead us to answer questions, our hypothesis, how to write a proper hypothesis, talking about an experiment, what are some of the controls and experimental aspects of the questions that we use to answer something, analyzing it, and then the conclusion. So all these are going to be parts of the scientific method. So with an observation, we're going to be using those 5 senses we have, right? Sometimes you might use different tools to enhance those senses, such as a microscope for being able to see those type of things, right? But these are all going to be what we use to be able to answer questions and kind of get an idea of what our initial concepts and questions are. Here, this is a picture. This is an image. You see a cat. You see some water. You probably can't tell, but this is a puddle of water the cat's in. And then you see this knocked over vase. Really important to make sure that we don't confuse our observations with interpretations. Big, big, big mistake. So if I said, what's the observation in this picture? What would you say? Would you say the observation is the cat knocked over the vase? No, that's an interpretation. We see a cat in a puddle of water. That's an observation. It's very objective. We're not putting blame on the cat, right? We're not taking our experiences of not liking cats maybe and coming up with an answer. We're really just looking at exactly what we see. Another example is about observations seen here. So this is another image we can look at, and then we can actually spend a little time in class focusing on this same picture and hopefully trying to identify observations versus interpretation. So I'll give you kind of a head start to look at it, but we'll spend more time on it in class. So with that scientific method, let's say I had an observation. I noticed that students who spend more time studying tend to do well, right? If I'm walking in the building, I say, wow, you're still here. It's late at night. You're studying still. Oh my gosh, look, you got an 85 on your test. You did pretty good. That's what I noticed. That's an observation, something I can see, something I can start to get my gears rolling. So looking at the next step of the scientific method, we have to ask ourselves, how can we answer this in the appropriate manner? The way is by looking at a hypothesis or creating a hypothesis. This is going to be a proposed explanation. Sometimes when people describe it, they say an educated guess, which that's not the best scientific way, but the educated part means we're actually pulling from previous observations, right? An educated guess is when there are clouds, it's going to rain, right? That means if there's clouds, it's going to rain. Where are we pulling this from? We're pulling this from observations. Every time I see a cloud, it rains. We're pulling this from information, right? We know that clouds are contributing to rain. That's where rain comes from. So that's where all those things are going to come together. When you have a hypothesis, it must be 2 things. One is testable. The other one is falsifiable. What does it mean to be testable? It means you have to be able to test whatever you say. So if you're making a hypothesis, and your hypothesis is that if I throw a ball into the ocean, it will travel all the way to the bottom. the ocean is very, very hard to track the bottom. It's a very, very deep bottom. There's no way to test if something made it to the bottom, right? It's just not possible. So that's an example of something that has to be testable. Falsifiable means we have to be able to prove it to be true, which kind of goes along with testable. So if I say something is falsifiable, that means if I tell you a secret, then you won't think about it throughout the day. There's no way for me to truly test if you think about something or not, right? I can't go into your brain and count the amount of times that you think about something. It's just not able to be proved to be false, right? So those are examples of testable and falsifiable. With the hypothesis, it contains kind of a special formula, and it's going to include a specific action, right? Something specific followed by something measurable, right? We usually use the formula if something specific happens, then something measurable happens. So let's say if I throw a ball at my dog, then he will jump. Now, can I throw a ball at my dog? Yes, that's something I can specifically do. You can see me throw a ball at my dog. Measurable. Then he jumps. Is there a way to see if something jumps? Yeah. If this is the dog, sorry dog, I'm drawing a bad picture of a dog. If this is a dog and this is the ground, I can see if the dog is jumping or not. I can measure if it jumped, right? Those are kind of things that we're looking for in a hypothesis. So let's say if students study 1 hour a day, can I measure that? Yes. Is that something specific rather? Yes, specific. Then they will pass bio 102. Can I measure if they pass bio 102? Yes. Pass, fail. Really simple. With the experiment, this is going to be a number of different procedures that are designed to test that hypothesis. So there are some key words I want y'all to understand as it relates to your experiment. One is your control group. Your control group is going to stay the same, right? It's going to stay the same. It's not going to be something you change. I'll show you an example on the next page. The experimental group is going to be the variable that you change, right? Let's say, if I say eating strawberries, something along the line about if I eat strawberries, then I will lose weight. If I eat strawberries. Sorry, my grammar is a little off. This little freestyle comment. The control group is going to stay the same. So if we have two groups of people, one group, no strawberries. That's the control group, right? That's going to be the group of people where they stay the same. The experimental group is going to be the one where we change one variable. What's that variable going to be? Eating strawberries, right? They're going to be the different group. The independent variable is another term that we want to introduce. This is going to be controlled by the investigator. So this is going to be the offering of strawberries. The fact that I'm giving you a strawberry, that's going to be my independent variable. Some groups get strawberries, some groups do not get strawberries. My dependent variable is the thing that's measured. So what's measured in the case that if I eat strawberries, then you will lose weight? Their weight is being measured, right? That's an example. Another image here of independent and dependent. The variable that we change will be the amount of water, right? So this is probably an example about, the amount of water that we give a plant, how does that affect its growth? The water itself, the amount of that water, is going to be the independent variable. The thing we're looking for is maybe the size of the plants, the number of leaves, is it alive or dead? These are just different kind of components that we might be looking at. In this particular case, the control group would be the plants with maybe no water, The experimental group will be the plants with, 20 ounces of water. Another example is here, really similar concept. Again, control group, no water. Experimental group, plus water. Independent variable, the thing that we control, the amount of water. You either get 20 ounces, you either get 0 ounces, right? Dependent variable, the amount of seeds that sprout. What are we measuring? So, in my opinion, this slide here is the best slide to give you an understanding of independent variable, dependent variable, experimental group, control group. So this is the one you want to stick with if you're having trouble understanding it. Looking at this slide is going to help. So then with our data, at the end of an experiment, we are going to get data. Data can either be quantitative, so look at that fourth letter, N stands for numerical, quantitative, or it can be qualitative. Think for L as, I always thought of L as looks, right? Something that's descriptive. So example, quantitative data will be something like the plant made 7 new leaves, right? It's a number. Versus qualitative would be the plant was dry and brown, right? That's going to be something you can visualize. It's not a number though. And then in science, we use a lot of statistical significant numbers to be able to understand how important something has been. So again, if we did this experiment, control group, no studying, experimental group, 1 hour studying. Let's say we analyze it. Wow, look at that. Students who study for 1 hour daily were 10 times more likely to pass the class. At the conclusion of this, we're just going to analyze and interpret our data. What does it mean? And finally, does it support our hypothesis or does it reject our hypothesis? In science, we can communicate this to other scientists and the public through publications, through presentations. We know a lot about this, especially in COVID era. We're really familiar with how scientists communicate. Also in science, sometimes we use model organisms. That means instead of doing all of our studies on humans, we might do our studies on other organisms. So think about why we might study a mouse or a fish instead of a human. This is because sometimes we don't want to damage humans. If we're trying to understand a disease, it will be really challenging to give it to healthy people because now they're susceptible to that disease. So there are different types of organisms we can use, and I can talk about that in depth if anyone interested in research. And again, science is not a rigid process. I feel like 2020 has really shown us it's not rigid. There's a lot of loops. There's a lot of back and forth. You start here, you go all over the place, you end up, one place here, and another time you end up here. But it doesn't mean it's wrong. It just means there's a lot of different ways to go about things. And sometimes if we get new observations, right, that's going to change our hypothesis. So think about some of the challenges we've had with COVID. I think that's why, as a community, people don't really trust some of the science, but it's because as we get new information, that changes the experiment, which changes the results, which changes what we tell people to do to be safe. So these are things I want y'all to really consider. This is very, very relevant, especially in our day and time now. Okay, we're actually going to skip ahead on this slide. So if it's in your notes, just go ahead and skip on it. We won't cover this one too much. We will briefly talk about the characteristics of science, and then we will be done. So scientific knowledge is theory-based. So a theory is going to be some type of accepted explanation of the world. And it's usually based on repeated observation and repeated experimentation. So think of a theory you may have heard of. Maybe it's, the Big Bang Theory. Maybe it's the theory of evolution, I think there's an atomic theory. All these different aspects are going to be different theories you've heard of. But I want you to focus on the characteristics of a theory. Widely accepted. Everyone says, hey, this is what's happening. Never 100% proven. We can't go back in time and see how the earth started. That's not going to happen. And then they can be modified over time. Again, we're talking about experimentation and observation. So the more we can get more experiments, we might be able to shift those theories in essence. So think about theories. Sometimes you hear A lot of scientific theories are well supported by data and testing, but then you may have heard some non-scientific theories. So a great example is, different things on the market. Maybe things like, tummy tea or certain types of medications or vitamins or skincare line. You might see things that say this is not based on facts or careful study. This information is not confirmed by the FDA. All these different things, some of that can be based on anecdotal evidence. So you just want to make sure that you're not being subjected to people think they're giving you science, but it's not the real deal. Also, with these non-scientific theories, a lot of them are based on personal experiences, right? Like, I have a theory that when it always rains when I have plans. Like, of course it's not going to happen. That's not something you've actually tested, probably. But that is just a concept of a non-scientific theory. It's not based on repeated experiments. So, as we kind of close up, I talked about this in the very beginning, and I'm going to come back to it. Science is objective. If something is objective, that means it actually displays skepticism, which means that you question things. You say, is this correct? I'm taking my opinion and my personal ideas out of it, so I want to question and ask and make sure that things are being done correctly. And we also have a lot of mechanisms in science that help people to cross-check their work. So for instance, if I wanted to publish something, a paper about scientific fact, I have to have other colleagues in the field who know what they're talking about give me the thumbs up and say, hey, this is correct. This is not a lie. This is not fake data. This is real data. So we have a lot of cross-checking. That's what makes science so great. Subjective is where it has personal values, beliefs, feelings, trainings. And unfortunately, science has the ability to be both, right? Why is it subjective and objective? Well, because the people running experiments are people, right? They're people, normal people. So they may have had certain types of values, beliefs, experiences, intuition, training, all these different things that make them look at a question a different way. So that's why it's so critical that we try to bring a sense of diversity into science, because we need people from different perspectives and different walks of life to be answering these questions. So this concludes the slides for this particular chapter. Please rewind it if you had any questions, and if you have anything else you'd like to discuss, we'll talk about it in class.